Queen Nerdling here. Today we're going to be discussing how substances get into cells and why these guys are so small. So let's go ahead and get started. So specifically, we're going to be discussing the plasma membrane and the properties of the plasma membrane, as well as why cells are so small and why there are limitations to cell size. So the question that you should be asking yourself is, how do cells establish and maintain their internal environments that are different from their external environments? So first of all, the cell membrane is the boundary between the cell and its environment, and it controls the transport of materials both into and out of the cell. So as we discussed in previous lectures, that plasma membrane is composed of a phospholipid bilayer. So we have our phosphate head. This is hydrophilic or water-loving, and it is also polar, meaning that it has a charge. And it's also composed of these fatty acid tails. Now the tails are hydrophobic or water-fearing, and they are non-polar and lack a charge. And they're arranged in this little lipid bilayer with our phosphate heads on the internal as well as the external surface of the cell, and our lipid tails, which are going to be internal. So our phospholipid bilayer is also referred to as the fluid mosaic model. Now fluid because there is actual motion, and mosaic because scientists thought it really looked like a piece of art because of all of the different components that compose that plasma membrane. So we do have the phospholipids, which are amphipathic, having those two charges, and they will simultaneously actually form a bilayer. So if you were just to kind of plop these together, they would actually start to develop out these phospholipid bilayers. So again, they have those hydrophobic tails on the inside, and we have our hydrophobic heads that face the outside as well as the inside of the cell. Now the mosaic component, again, the bilayer is going to be embedded with all kinds of different proteins and different types of fats, as well as carbohydrates. And it gives it this little, mosaic look to it if you were looking at it with a microscope. So cholesterol actually gives the bilayer its stability, and the cholesterol is kind of integrated throughout that plasma membrane. So as far as other components of that plasma membrane, we have glycolipids. Now, if you kind of break that apart, glyco is going to be referring to carbohydrates. So this is a combination of lipids and carbohydrates that have formed this glycosidic bond. And then we also have glycoproteins, which is the same concept, except now we have our carbohydrate that is attached to our protein. And this can flow around the surface of the cell, as well as going to traverse the membrane itself. Now these glycoproteins actually play a very important role in cellular communication. So why are all of these proteins the perfect molecule to build structures in the cell membranes? Well, because they have different charges and different properties. Now, if you remember, if we're talking about monomers, proteins are composed of monomers called amino acids. And amino acids have their own properties. So looking at these amino acids, thinking about what they all have in common, all of these amino acids on the screen right here are actually non-polar, meaning that they are hydrophobic. Now, do not worry, you do not need to memorize all of the amino acids and know what their charges are. You should be able to look at the amino acids and kind of get a feel for what the property of them might be. So the same thing holds true to these amino acids. If we look at all of these, one thing that they all have in common is that they are polar and charged, meaning that they are hydrophilic or water-loving. So the proteins themselves are anchor molecules. So within the membrane, we have non-polar amino acids, which are hydrophobic, and these help to anchor the protein inside the membrane itself. And then on the outer surface of the membrane, we have polar amino acids, meaning they actually do have a charge. Now these are hydrophilic, and they extend into that extracellular fluid as well as the cytosol itself. And so you see this protein is basically traversing that plasma membrane, and it could serve for cell signaling. Now, looking at different examples of proteins and how they're integrated into the plasma membrane, as well as what function or role that each of them play, we have some examples of aquaporins, aqua meaning water, and pore is basically saying kind of like a little tunnel or a channel that goes into and out of the cell and allows substances to move into or out of the cells. So this water channel allows for bulk movement of, you guessed it, water. And then we also have protein pumps. So these are going to allow charged molecules or ions to pass through that plasma membrane. So this is actually part of the electron transport chain in photosynthesis and cell respiration. And this is actually necessary for generating ATP, which, as you know, is basically the molecule that fuels every single metabolic reaction that goes on in our body. So there are so many different examples of proteins and how they're integrated into that plasma membrane. We have transporter proteins. We have proteins that serve for enzymatic activities and functions. We have proteins that serve as cell surface signaling receptors that are actually going to receive signals that are transmitted throughout our body. We have different cell surface identity markers, and these are very important when we get into talking about immune system and how cells recognize good versus bad or antigens. We also have proteins that function for cell adhesion and attachment to the cytoskeleton to serve for structure. So the membrane, because of all of the properties, it makes it selectively permeable, meaning that only small non-polar molecules can move through that bilayer. So some examples are oxygen and carbon dioxide. Now, there are a few exceptions to the rule. So we can have small polar molecules like water that move in very, very tiny amounts through that plasma membrane. Now, our large and charged molecules have to move through the protein in pores or channels. And some examples of those would be glucose and amino acids. So cells having to take in glucose or amino acids to help to build different structures. Now, looking at this little animation, we have our cytoplasm right here, which is going to be internal. And then we have our external environment, and this would be our plasma membrane. As you can see, those smaller water molecules can move freely across that plasma membrane. However, when we have water molecules that are going to be attached to a larger macromolecule, those cannot move freely through the plasma membrane. Now, this is also a demo of a topic we're going to get into quite soon, which is going to be the passive diffusion of water, or osmosis. And you see the water moving from a higher water potential down that concentration gradient into a lower water potential. Or if we wanted to talk about the actual substances, the water is going to move from a hypotonic solution towards the hypertonic solution, which in this sense is going to be internal to the cell or in that cytoplasm. So we also have cell walls. Now, cell walls are found in bacteria as well as some eukaryotes, including plants and fungi. So most of the time, these are inactive structures and they serve for support. And in some cases, they serve as a permeable barrier. They're composed of complex polysaccharides, poly meaning many, and saccharides is referring to our carbohydrates, specifically sugars. So in plants, the cell wall is composed of cellulose, and fungi, the cell wall is composed of chitin, and in bacteria, the cell walls are composed of peptidoglycan. So the next question that we have is, how does cell size affect the ability to function and transport materials? Now, cells have both an upper and a lower limit. Lower limit meaning having enough to actually create a single-celled organism or life. So we can't go too small, or we can't actually put all of those components together to form a fully functional cell. So we have very teeny tiny bacteria and the smallest mycoplasmas, which are about 1/10 of a micron to 1 micron in diameter. And just to kind of give you reference and provide you, so a single hair on my head, the diameter would be approximately 20 microns. And a micron or a micrometer, so looking at this symbol right here, is 1/1,000,000th of a meter. So super teeny tiny. So most bacteria fall into a size of between 1 to 10 microns or micrometers. Now, when we get into our upper limits, meaning as large as cells can possibly be, where they still perform their function at a good rate and can conduct all of those metabolic processes. So the upper limit of that is going to be about 10 to 100 microns in eukaryotic cells. So there are limits to size. So we have metabolic requirements that set that upper limit. In large cells, we cannot move enough material into and out of the cell fast enough to support life itself. So how do cells get bigger? Well, they start to become actually multicellular organisms and the cell starts to divide. But this in and of itself has different problems because now we have a bunch of cells together and those cells are going to form a tissue. And tissues have different needs or different metabolic needs depending upon what type of tissue it is and what that tissue's function is. So again, we have those limits to cell size. We have the lower limit and the upper limit. That lower limit is due to the basic materials that are required to be a functional cell. And that upper limit is basically put into check or regulated by the surface area to volume ratio. Now, the higher the surface area to volume ratio is, the better exchange of materials and the faster those cells can actually exchange the materials. Now, as the cells increase their size, that cell surface area to volume ratio decreases, meaning that the volume of the cell grows at a much larger rate than the surface area. So that's why we can't have these really, really large cells. Now, in your classes, and I know in my classes, I always do a cell size lab where I give my students these little neon pink agar cubes, and they have a really teeny tiny one, they have a medium-sized one, and they have a large-sized one. And we're looking at diffusion rates. So they'll put these little bright pink cubes, which is because they have phenolphthalein and sodium hydroxide reaction. We'll talk about that later. And they'll put them into an acid. Sometimes I use vinegar, and sometimes I'll use a weak hydrochloric acid solution. So they'll put them in, set the timer for 10 minutes, and then they have to measure the diffusion or the amount of pink that has basically diffused out of the cell. And so what they will find is the same distance has been traveled. So that white edge that was once pink is the same for both the small, medium, and large cells. Now what they find is that obviously it would have taken a lot longer for that diffusion to occur into the middle of the largest cell versus the smallest cell. And so that's a concept that my students tend to struggle with a little bit because they're like, well, it diffused faster. And then we have the conversation of it didn't diffuse faster, it would just take a longer amount of time. And the example that I like to use is if I decide I'm gonna hop in my car and I live in Houston, Texas. So for those of you watching this, you aren't from Houston, it's gross, it's hot, it's humid, so you wanna kind of move. So I'll hop in my car and I'll say, I'm going to go to Austin, which is about 2.5 hours away. I'm gonna drive 70 miles an hour. So it's gonna take me 2.5 hours to get to Austin, Texas, driving at 70 miles per hour. All right, so speed. Now, if I decided I want to go visit my parents in my hometown of Truth or Consequences, New Mexico, and yes, that is actually the name of my town, Truth or Consequences. So same concept, I would get in my car, I would go 70 miles an hour, except this time I'd have to travel almost 900 miles. So that is going to take me about 13 to 14 hours. So I'm going the same speed, whether I'm going to Austin or whether I'm going to Truth or Consequences, New Mexico. So that speed or the rate is going to stay the same. However, the distance that I have to travel increases. As the distance increases, so too does the time it takes me to get there. The same concept holds true for cells. The larger the cell is, the longer it will take materials to diffuse into the nucleus and out of the nucleus into the cytoplasm and eventually out into the extracellular fluid. So I hope that was helpful. For more videos like this one, you can hop on to www.nerdlingscience.com. This is the Queen Nerdling signing off for today. I hope everybody has an awesome day.